So as Smith introduced, I'm Manuel Martinez from UPC, and I'm going to present our service developed within the iMagine project, which is fish detection and classification at EMSO-OBSEA. So what are we going to cover in this presentation? First, I'm going to start about what is ecosystem monitoring and how we want to do it with artificial intelligence. Then we will continue to some very brief review of what is object detection. So object detection in a nutshell, how we prepared our data, how did we train it, which metrics are we using to evaluate it, and how we can fine-tune the models. And finally, I will present some scientific results that we achieved. Okay, so I'm working at OBSEA, which is part of EMSO-OBSEA. So what is EMSO-OBSEA? Well, EMSO-OBSEA is a distributed infrastructure. So it has 15 underwater nodes, meaning underwater stations, fixed point observation systems, which are devoted to monitor the oceans and its evolution in different domains, such as physical oceanography and geochemistry, and also biological monitoring. So on the right, you can see the map of the different facilities scattered across European waters. And I'm working here at OBSEA, which is in the Catalan coast in Northwest Mediterranean Sea. And these are some pictures of OBSEA. OBSEA is a cabled observatory, an underwater cabled observatory, meaning that we have a power and communication cable from the shore station to this seafloor node where we have a lot of sensors deployed that can monitor different kinds of parameters. We monitor temperature, salinity, pH, waves, currents, sound, and we also have some cameras. So we are monitoring the biodiversity and the ecology in the area based on underwater images. So over the years, we have deployed many different cameras. We started, we have some pictures from 2011 until now. At the beginning, it was very low quality cameras because they were old models. It was quite some years ago. And recently, we have deployed new HD cameras, as you will see, much better. And we have been doing fish abundance and behavior studies of the fish community in the area. And basically doing it by counting fish, to take pictures with the camera and counting fish one by one. As you can see in the bottom right, we have in the circle is the camera, which is pointing to this big concrete structure, which is called a biotop, which is basically an artificial reef, where fish hide there, and there's a lot of biodiversity in this artificial reef, and we are monitoring the changes over time in this fish community. Okay, so how was traditionally done this ecological monitoring or this ecosystem monitoring? How has it been done traditionally? So basically, you need some underwater pictures, and traditionally scientists and biologists took these pictures and counted fish manually, one by one, in every picture, and then put all this information in, for instance, an Excel spreadsheet, and then they could create some abundance time series to see which kind of fish appear in each season, etc. Of course, this is very, very time consuming. It requires a lot of manpower to take the images, count fish one by one, and write them down into a spreadsheet. And of course, this can be automated. And this is exactly what we have been doing in the iMagine project. We have developed this service for AI-based ecological monitoring, where we take the same pictures, but instead of having a scientist that is manually counting all the different fish specimens, we have an AI model that automatically produces some detections, and we store these detections into a database. So the abundance time series that we can produce out of these pictures is fully automated without any human in the loop. So the traditional ecosystem monitoring is very time consuming, not reproducible because not everyone sees the same amount of fish. It's not as simple. You need expertise in biology because, of course, you need to know the taxa of every fish, and it's an extremely repetitive task. It's very boring. It takes a lot of time. So, and it's a waste of human potential to have someone with biology expertise looking at thousands and thousands of pictures. So, on the other hand, when you have an AI-based ecosystem monitoring, of course, you will need some IT skills and a lot of effort to set the system up. But once you have the model trained and you have your service up, it's a fully automated workflow without any human in the loop, and you can automatically analyze thousands and thousands of images without human intervention. And then you can get this objective that we are working, which is the automate the ecological monitoring. And we can have scientists doing actual science instead of counting fish in pictures. Okay, so what are the steps in an object detection application, in this case for fish monitoring? First step, of course, is to collect pictures, like the ones here. Then you need to label the data, because we are going to transfer our knowledge about fish to a machine. So we need to tell to the machine what is a fish, what kind of fish it is. So we are doing it by labeling the data. Then we need to select a model. There are a lot of different AI models out there. We need to select one. We will need to format the data to match this model. Each model has its own specificities, and we need to format the labels, format the files to be able to allow the model to understand it. And then we need to train the model with the data. This is the step where the knowledge that we put into the picture and the labels is transferred to the machine. And finally, once the training is done, and if everything went as planned, then we can use this model to do inference. So basically to do predictions of fish or any kind of object inside your pictures. Okay, to prepare a data set, I'm not going to enter into the details, but there are a lot of labeling tools and some issues that you may encounter, especially in underwater pictures, is image quality, specifically biofouling. So any kind of leftover dirt in the glass. So underwater environment is very harsh, and you can get a lot of dirt and a lot of, well, which it's actually live, but it's not the life that you want to monitor. And it can get into your optics. So it can be really hard to have good quality pictures. So it's something that you need to take into account in underwater. And here we have an example of a lot of dirt in the glass being detected as fish, which of course is not good. So it's something that you need to be careful with your data. Another important aspect is that you need to label everything, because for instance, here we have a very clear fish, very nice one in the front of the picture, but we have a lot of smaller fish hidden in the background. And when you are labeling your data, if you are not labeling everything, then your model is going to confuse this background fish with the just general background. So it's difficult to, you have to label everything, which is not always easy, because sometimes not even the human eye can distinguish which kind of fish it is. Another thing that we need to take into account is the balancing. So balancing it means that we need to have almost a comparative amount of specimens of every class, every fish in this case, to avoid underrepresented classes. So if we are training with cats and dogs, we have 10,000 cats and only 2 dogs, of course the model is not going to perform properly. And this is what happens in our case. For instance, take this picture. We want to, we have a lot of the small fish, the chromis, which are very, very abundant. They're the smallest one, the most abundant ones. So we have a lot of overrepresentation with these classes. And if we select pictures with these zebra-like fish, the other ones, the orange ones, because we want to improve the presence of this in our data set, at the same time, you get more of the chromis one. So it's difficult in this particular scenario not to have underrepresented classes. And as you can see here, this is an old snapshot. Now we have more pictures, but this is a few months ago what we have in our data set. And as you can see, we have thousands of chromis, but very few other fish specimens. That's life. I mean, that's how the quest system works. You have a lot of small fish and less bigger fish. So what we decide to do is to like split this data set. We have some of, and not to include all the labels into our model, because if you have only 10 or 15 labels, it's for sure it's not going to work for this class. So we have this sort of Solomonic decision where we, at some point, we cut the data set and we say that we are going to exclude some of the classes of the training because we are going to get worse results with these classes. For model selection, well, there are a lot of AI models out there. It's a mature field, rapidly evolving. There are a lot of applications out there, very interesting. If you want to set up your own model, object detection or any kind of AI-based system, and you don't know which model to choose, I recommend you to browse through this Papers with Code website, which has a lot of reviews of different tasks and different algorithms, and it's very useful. And in our case, after delving a bit, we selected the YOLO model, which it has because it has a good balance between precision and speed. It's quite easy to use, and it has a very good documentation. So now it's time to, we have the data, we have the labels. So how can we train our data? So first we need to format the data. Yes.
Just how do you want to handle questions? Do you want to take the question if it's already here, or do you want to do all the questions at the end?
I think it's better to have them at the end, I guess.
Okay. Okay.
Because probably I'm going to answer some of the questions during the slides.
Sure.
Okay, so you need to format the data following the documentation of the model, and then just train the model, usually just a command that it will take hours or even days. But then you let the machine work. And finally, you get your model that you can use for inference. And now it's, okay, we have a model, it's working, but now we need to assess how good is it? I mean, meaning what kind of metrics and objective measures do we have to see how this model is performing. So I'm going to go very quickly through this. I'm not going to get into the details, but there are several metrics like intersection over union, which is how good your prediction are when they are compared to the labels, the precision, the recall, meaning it's a balance of true positives, false positives, etc. So I will leave it here if anyone is interested, but it's not going to go into the details. Then you have average precision, where you combine precision and recall. Then you have MAP, which you average all of these metrics. So there are a lot of metrics where to understand how good your model is. And we are mainly using mean average precision to compare our different trainings. So I think it's not interesting now to go into the details, but we are now close to 0.9 MAP. Okay, now once you have a model trained and you want to fine tune it, to improve it, there are several things that can be done. First thing is to increase the image size, because it means image quality. We have here the same image in different resolutions. Of course, if we are using a very low resolution, is something like the picture in the right, which is very difficult to understand what is it, even for a machine. Of course, if you have a high resolution picture like the one in the left, it's simpler for us and for the machine to understand. Of course, the higher the image size or the resolution, slower is going to be the training and the inference, but you will have better results. Epochs, meaning how many times is your model going through the data set. The higher the better, until it stagnates into a point, and then you get the best, something usually between 100 and 200. Batch size is maybe also important, but then you need more IT resources. And usually the best way to improve your model is to have more training data and well-defined training data. Okay, so I was speaking about general object detection. What about our use case? So we did all these steps. We have pictures. We labeled a lot of data, even with some classes that are not included in the model. And this data set is published and available in Zenodo. We selected the YOLO model. The results that I'm going to present are based on YOLO v8. Currently, we are working on improving it to YOLO v11. And this will be released soon, I hope. We formatted the data, meaning we selected some of the classes, split it into training, validation, and test. This dataset is also available in Zenodo. And the results of the training, so meaning that the trained AI model that can be automatically used, is also published in Zenodo. So feel free to download all this data and to use it. It's for free, it's available. Just please, if you use it, cite us. So this is just a quick overview. So I presented a lot of things, but if you want to replicate our work, what are the steps? So it's not that complex. I mean, to have a first model running, you just need to install the Python package with pip install ultrarate is the package that contains the YOLO model. You can download our dataset and unzip it. You will need to edit your the configuration file to put the paths to the data that you downloaded. So just the path to the zip file. And with this command, YOLO tasktm mode train, you will train your own model with our data. So you should get the same results. So if anyone wants to try, here are the steps on how to train the model with our data. To improve, once you have a first model, if you want to improve, you can have semi-automatic labeling. This means that instead of labeling manually all the images, which takes a lot of time, you can use a trained model to give you some predictions of new pictures. And then you can just drag and drop these pictures into your labeling software, and then you can manually adjust it to be sure that they are 100% correct. You can retrain, and then you have this improved model and you close the loop. So once you have the first model, this model can help you in the labeling of new images. So once you have a first model, it's much simpler to get more pictures with better labels and retrain it. So that's something that can also be done. Of course, this is a part of the iMagine project, and we have been using the iMagine platform to train. So we have dedicated resources for training. We also have some off-the-shelf AI models, a lot of things that are published in the website, in the marketplace, with all their code published in GitHub. Mainly, I think that all of them are Dockerized, so you can just pull the image from Docker Hub and download it. So if you want to, you can just go into the iMagine website and check the marketplace, and you will see you have not only this model, but a lot of different models that can be used for your own applications. Okay, so let's show some results. This is actually an old video, probably. Okay, I can make it. This is an old video that was one of the first tests. It was quite okay. With just 400 pictures, we could have something like this. So we'd run the inference on a video offline, and then we have predictions. So it detects in every frame all the fish and all the species. It's quite good. We have improved this, and now we have real-time video. We set up this, now it's live. It should be working in real time, hopefully forever. We will see. And if you go, you can scan the QR code, or you can just go to the link, and let me see if it should be working. Okay, and this is live now. Probably you are not going to see it very smoothly because of Zoom is not transmitting all the frames when I share the screen. But if you go to YouTube and search for OBSEA_AI channel, you should see this in real time. And as you can see, we are running this on a dedicated machine. And well, we have predictions for all the frames, and it actually works quite well. It see fish that even I don't see usually. And well, so we want to, this is mainly for dissemination. It doesn't have a lot of scientific, let's say, applications at the moment, but I think it's very nice to see the work that we have been doing. Sometimes you see some frames change the color, which means that sometimes it can get confused depending on the orientation of the fish, but most of the time it works quite well. Okay, so let me go back to the presentation. So again, if you want, you can just scan this QR code and you will see this real time. Okay, now some scientific results. We have been, we applied these algorithms into our data archive. So the idea is to have long-term ecological monitoring data time series with different fish. And we have data since 2009, but we could not yet have predictions of all the pictures, mainly because of with the low quality issues. We are working on it. But what we do have at the moment is all the HD cameras that we have deployed from 2023 to now. We have time series for all these cameras, and it's available on Zenodo. And here on the bottom right, you can see the time series from January 2023 until I think 2 days ago, when I exported the dataset, with at the top the total number of fish, and at the bottom the number of fish, the fish disaggregated by species. And this can be used for any kind of biological studies to check, predator-prey interactions, abundance, where and at which time of day or which season does some species appear. But this is just for the HD cameras, and we are working with low quality cameras, and hopefully we will publish this data very soon. We also have some results for the Slack Reef project. The Slack Reef project is it's a side project that we have. It's not related to iMagine, but we are using the same technologies. This project, the main aim is to produce 3D printed artificial reefs, mainly using waste from the steel industry, which is called the slag. So instead of just disposing this waste, we want to use this. We are using this waste actually to create these puzzle-like pieces that can be piled together to create artificial reefs. And why do we want artificial reefs? Mainly for two reasons. One is to protect the ecosystem, and the other for shoreline in bigger deployments. So in small deployments, when you have degraded areas without reefs, usually the small fish doesn't have anywhere to hide, and they are quickly predated. So when you put a lot of this kind of pieces, you create artificial reefs, and you see a lot of small fish go there to hide from the predators. And then you have a more healthy environment, as we will see in a video later. And when you have big deployments, like thousands of these deployed, it can even protect the shoreline, because when you have some artificial reefs, well, when you have reefs or artificial reefs, in a lot of places, like in our area, there used to be reefs, but due to overfishing with trawling, they get completely destroyed some time ago. And now there are experiments to restore them artificially. And this can help in cases of huge storms. If you have a lot of these reefs, they absorb some of the energy of the storms, and then the shoreline is not affected as much as it is right now, where we have every year we have a lot of issues due to in the shoreline with the storms. The data is published in Zenodo here, and again we have the time series on the bottom. And here, let me show you a video. Let me see if I can put this in high quality. Okay, and as you can see, the small fish are hiding inside these artificial reefs, while the big ones, the predators, are trying to catch them. And if you look in the surroundings, you don't see almost any kind of small fish, because they are all hiding. So that's why the reefs are so important in the ecosystem, because they protect the small fish from being eaten. And in the long term, this means that they have a more chance of surviving. And if all the big fish, if all the small fish are eaten, then the big ones starve. So what? This is another of the experiments that we did. And the last, next slide, okay. And the last one is a series of dolphin carcass experiments that we did, a couple of them. So from time to time, dolphin carcasses are found on the coast. Mainly they are killed by fishermen because they are entangled in the nets. And we ran a couple of experiments trying to see what happens when one of these mammals fall into a coastal area. So the scientific objectives are to see the ecological changes and also to determine which species have scavenging behavior. So it was in our area, it was found a stranded dolphin, and we took it and we placed it in front of the camera to see the changes in the ecosystem. And here you have a time lapse of the poor dolphin being eaten by all the fish. And you see the cycle day, night, and with all the fish starting to eat the dolphin. And of course, in the bottom again, you can see all the time series. We did a couple of these experiments, and the data is also available in Zenodo. So, and now that the dolphin is gone. Okay. So if you want to, a lot of fish eating the dolphin. And if you want to get the data, again, you just scan the QR code or just go into Zenodo and look for OBSEA or iMagine. It should be in both communities and you can just download the data. Okay, the last service that I'm going to present that we did is we have all this data integrated into our infrastructure. So we have an API for machine-to-machine interactions where anyone, because the API is open, can query for picture, for inferences, for fish detections. It will you can have we have in the right an example which is just standard JSON API response where we have the taxa detected, you have the confidence of the detection, you have the bounding boxes of the fish. You can even have the link to the original image, the link to the processed image with the bounding boxes. So if anyone wants to have this data in real time, it's also possible. It's freely available. Anyone can use it. We are using SensorThings API, which is a standard API for IoT, just to query the data. We are exposing all the pictures, the raw files and the processed files using an engine service. Everything is connected. So everyone with average IT skills could set up a system to gather our data in a few minutes. And well, we periodically export these to Zenodo, but we're not doing it on a daily basis. Probably we're going to do it every year or so. So if anyone really wants to have the latest data, real-time data, please just you can send me an email. I will explain. I can send a lot of documentation on how to use these APIs. Okay, so that's all. I just want to put here a list of all the data sets and resources that we have been producing within the iMagine project. We have mainly two data sets for training, one with the raw pictures with the labels. So because you can check and do whatever script you want. We have the training data set where you can just download and train with a couple of commands. So it's prepared to train a YOLO model. We also published the YOLO V8 model that we are using for inference. And we published 4 different scientific data sets with the inference. Fish detection of OBSEA, like the big biotop that is there, that has been there for decades. The small artificial 3D printed reef, which is called the slug reef reef. So we also have this dataset published, and we have also published the 2 different experiments with the dolphin carcass and the biodiversity associated to this. And of course, you can also check our live video on YouTube. Just a small comment, the links may change over time, so be sure to check our channel, OBSEA_AI. And that's all from my side. So, yes. We're working in new models because this never ends. We have more data, better data, we have better models. So very soon we will be publishing new AI models. And with these new AI models, we will publish new data sets. So stay tuned. We will have more and better data coming out soon. And that was all from my side. Thank you very much for your attention.